Hi, my name is Ingrid Mejia, and I'll be presenting Cisplatin. So, Cisplatin is used to stop the cell division, and the way it does that is by binding to DNA, and it interferes with the repair mechanism, and that is done by a molecule of water being able to replace a chloride ion component of cisplatin. The structure will then be able to bind to a DNA purine base. Another water molecule will usually then also replace another chloride ion on the structure of cisplatin and be able to bind to another side of a purine base. When cisplatin binds to these DNA bases, it forms DNA adducts. The adduct that has been created blocks transcription and DNA synthesis. The cell cycle is arrested, and if the DNA cannot be repaired, it will most likely end in apoptosis. Uh, so here I have a diagram on the right, and it shows the process of what I was trying to explain, how cisplatin enters the cell membrane, and then how the water molecule will replace the chloride ions, um, and then it will lead to binding to the DNA bases. So some common side effects that the drug has is nausea and vomiting, temporary hair loss, diarrhea, loss of ability to taste food, and signs of dehydration like dry mouth, um, and also some serious side effects that might develop later on through the process will probably be like loss of vision, hearing loss, and kidney problems. There's also some other serious side effects that might develop, like maybe when the patients might be vomiting, they will probably vomit blood, and it is advised that they should call their doctor, and also some other possible side effects as well, like pain in your eyes or something like that. So the history of cisplatin, it was first developed by Peroni in 1845 and was called Peroni salt, and structure was deduced by Alfred Werner in 1893. The mechanism of cisplatin in the cell cycle was discovered in 1965 by Dr. Rosenberg. It was discovered accidentally, actually. Um, Him and his colleagues have put E. coli in a solution with platinum electrodes in there, and as soon as it was turned on, they noticed that the cells had stopped dividing, and when they turned it off, they started dividing again. They had at first concluded that the electrical currents were the ones controlling the growth, and they actually spent 2 years trying to figure out how it worked, and they came to another conclusion that it was not the currents that were controlling the cell growth, but it was actually the platinum compound. um, Dr. Rosenberg later tested a cisplatin if cisplatin could block cell division in tumors, and he used a sarcoma mouse model for that. The mice were able to tolerate the cisplatin drug in low doses, but it was actually very toxic. The tumors shrank, and mice showed no return of tumors in that experiment. Um, the National Cancer Institute started funding clinical trials in human cancer patients in 1972, and successful clinical trials uh, led to the approval of the drug by the FDA in 1978. And it is now used in more than one type of cancer, not just testicular cancer. And it can also be used in lung and ovarian cancer treatments. And it was also combined with it can also be combined with other drugs as well. My question was that in what way can cancer cells still be resistant to chemotherapy drugs like cisplatin? And that concludes my presentation. Thank you.